Hello, we are going to go over your mitosis work from yesterday. This will suffice or be our note, I guess. We won't technically go through this in a separate note. So the work that you did previously will count as all the information you'll use about your understanding of mitosis, cell cycle, chromosomes, and so on. Okay. So the first little bit here was going to that website and just checking out the structure of chromosomes. It's not a huge amount of detail that we're going to go into, but we're going to give you the basic idea of how a chromosome forms, what it's composed of, and then ultimately what the chromosomes are going to be doing as they do serve a function and role within the cell itself. Otherwise, they wouldn't form, and they would just be chromatin the whole time, or even, I guess, not even chromatin, because the histones are all part of forming a chromosome. So if there was no chromosome formation, it would just be the naked DNA, which you could just draw very easily as far as a simple diagram goes. Anyway, so going through the questions here. So this is simply asking you to draw and then label the location or what a nucleosome is. And chromatin is DNA wrapped around histones, and each one of the wraps around a histone is we call a nucleosome. So this is depicting a single nucleosome where you have the histone protein, and then you have a certain amount of the DNA wrapping around it. And that's what makes chromatin. It has all these nucleosomes along it, and that's it. It doesn't go beyond that in structure. It's the amount of DNA that wraps around a histone, I guess, is it goes around once, almost two times. So it kind of goes around and then wraps and then goes off and it connects to more DNA or connects to another nucleosome and so on. So you have a long string of these along a chromatin molecule. Um, Now, base pairs or these BPs that I listed here, you don't really cover those in this course. It has to do specifically with the components that make up the DNA, which would be something you would do in grade 12, maybe in grade 11, depending on what the AP course is like for biology. So this is not so important, but it is presented in the where I guess where I found my information from. I don't know if it was listed in yours, but just a little heads up for the other unit there that's represented. Okay. Now, the chromatin and nucleosomes, what they start to do when a chromosome forms is they start to form into these solenoids. And what the DNA is going to do, along with the histones, it's going to form these kind of like ring like structures. Not continuously, this is not continuously connected to itself, but it does form this kind of ring where you could have something, I guess, in between going through the ring, so to speak. Okay, so it can fit through there. But this is supposed to represent, I don't know if that's going to show up very well, but you could look down this as a central spot there, and this nucleosome could go around my pencil or whatever. So there's like this empty axis inside of there, an open space. So the kind of repeating ring structure of these histones in DNA. So when they start to form into chromosomes. So the first thing that the chromatin does is it starts to wind up into these solenoids, and this is kind of held in place by the histones or helped by those proteins and so on. It's a fairly complex process. It's just that here we're just going to get the general understanding of it as opposed to all those smaller details. Okay, so this is looking down the length of it, and that's why it was drawn like this. Otherwise, you could draw it from like a side view if you wanted to, but you would probably only see three of these if you looked at it from the side. So if you were looking at it from this side here, if this was your eye here, you'd probably only see these three histones. You probably may not be able to see them if you looked at it from the side. Okay. Anyway, so what those solenoids do is they start to wrap around one another and they form these repeating, right? There's your six solenoids, six we're looking at from the side, but it's forming this kind of long tube. It's not solid walls or anything, but it's starting this structure of taking all the solenoids now and stacking them and stacking them and stacking them and bringing them close together and forming this greater structure, which is going to eventually be a chromosome. Okay? So I did make a note here saying that these are really large structures. Okay? And what's actually happening to the DNA is if you looked at your diagram that I gave you access, it gives you an approximate size of these. So I don't know the actual numbers here, but for instance, if this is measured as being 1 micrometer, right, that's pretty small. But when they start to condense, this is now 30 times bigger than that. And then when you get down to this one, let's just say that it's I don't know, let's just say it's 1,000. I'm making that number up. What's happening is you're going from very small structures, and when they start to come closer together, you get bigger. They're forming a bigger structure. And when they get to this point, they're going to get even bigger. It's probably a really big number, but you can always fact check it. But what's happening is as these numbers get bigger, that means that the structure itself is getting bigger, and you can start to see it. So when this number gets big enough, you can see with a microscope. And you'll never see these with your naked eye, no matter how big a chromosome, oh, I guess whatever chromosome forms, because it's still microscopic. But they are big enough to be able to see when they condense like this, and also have enough stand applied to them that you can see them when you're using a microscope. So the structure of the chromosome starts off from the chromatin, and then it starts to form another structure, and that structure builds upon another structure. And what you're having is this is called condensation, because you're condensing the whole long spread out piece of DNA into a more tightly knit structure, so it gets bigger in size. The length of it's still the same, and it's still the DNA is just a long, narrow molecule. But when you start to make these condensed structures, you take that very long thing and you put it into a shorter thing, but it's all squished together, so it becomes bigger, and you can start to see it. Okay, so that's the chromosome structure. And the next part of the diagram was once you have a chromosome, it does have certain parts to it. So after all those solenoids and histones and DNA condense and form these structures, there are certain locations that we're going to be concerned with for this course, and then there'll be stuff that you talk about later, most likely in grade 12. So over here, this is one chromosome, and this is supposed to be the sister chromatid of that chromosome. Now we'll get to that a little bit later when we get to the S phase, but for now you probably saw a drawing like this on the website, and then you could draw the locations that are important to understanding how chromosomes work. So the middle location is usually called a centromere, because centro sounds like middle or center, right? That's what that's kind of based upon. Now, not all centromeres are in the middle, but for all intents and purposes for this course, we'll just treat them as being in the middle. Okay. Now, the telomeres. Now, if you know about meiosis, there's also the telophase and telomere for this one, right? So telo would be kind of like the end, because the telophase is the end of the phase of mitosis, and this would be the end of a chromosome. So there's two ends to each chromosome, so you're going to have a telomere on each of those ends. Okay. The part in the middle that is also going to be part of the centromere is something called a kinetochore. Now, the kinetochore are where spindle fibers attach, and it looks like in the diagram that they might even produce a certain degree of spindle fibers there, or at least the proteins that the spindle fibers are made of, which are like intermediate fibers or microtubules and whatnot, but that's not for this course. Okay. All right, so telomeres are at the ends, centromeres are in the middle for all intents and purposes of this course. Now, the one confusing thing that we're going to talk about are the difference between a chromosome and a chromatid. Okay. Now, I made a little note down here that these chromatids are copies of one another. So as we'll learn later in the cell cycle, the DNA gets copied, which means essentially that each chromosome gets copied, because each chromosome is one molecule of DNA. Okay, but it gets copied, so that means that there's going to be a second copy of that DNA or a second chromosome. Okay? Now, we refer to these two copies of one another as being chromatids, even though our chromosome number doesn't increase, and we'll talk about the confusion with that a little bit later. Okay. Now, what I simply kind of think about is I think of chromatids about how many structures make up the chromosome. Okay. So in this drawing here, there are two structures, 1 and 2. So this would be like chromatid 1, and this would be chromatid 2, but they are copies of one another, so they are essentially the same chromosome. So we consider this copied as just being one chromosome. But that one chromosome is made up of two parts, which we call chromatids, chromatid 1 and chromatid 2. Now, when these separate, they will be by themselves, so they'll still be one chromosome, but since they only have one part to them, we call that one chromatid. It is confusing because the words sound the same. Okay, and it's only confusing because when you look at the genetic information, there's a stipulation that the chromosome number does not increase when it gets replicated, and we'll cover that a little bit later. Okay, but for now, this is collectively one chromosome because they would be attached together if they were copies, which we'll show later. And then there's two parts to this structure, so we call each part being a chromatid. Okay. All right. Now, the chromosomes don't occur for a non-specific reason. So cells generally live in a repeating cycle or a lifelong cycle, but this is just like a repeating cycle. I don't know the exact hours. You can Google how much time for each individual type of cell. There could be averages and so on. It doesn't really matter. Just think of this as a pie graph, and that the relative amount of how much of the pie these take up is how much time a cell will spend in it, given a day, a year, a month. We'll just say a day or whatever, something like that. Okay. So you can see that most of the time a cell spends in its day is going to be an interphase. Okay? And interphase is critically important because that is where a cell performs its functions. And your body and any living thing, because it's made up of cells, can't work without cells doing their functions. So if your cells are dividing, it's not going to be all of your cells at the same time. It'll be some cells some of the time, because if your heart cells are dividing, let's say, you can't have all of them dividing because your heart will stop functioning, because it can't divide and perform its functions at the same time. It's all or nothing, or it's all one or the other. So when you get to the other part of the cell cycle that's not performing its cell functions, you are in mitosis. And this is when a cell divides. But when a cell divides, it focuses entirely on that. It's a complicated thing for the cell to do. It can't do anything else at that time. So it's going to transition from functioning and then dividing, and then going back to functioning again and then dividing. And in that way, uh, since all your cells are not doing one or the other all at the same time, you'll have cells normally functioning like a heart, and then some of them will divide. And then when those ones are done, maybe some other ones will divide, and so forth. Okay? So this is a transitional thing, and not all cells are doing the same thing at the same time. Okay? So it's broken down into these different parts, which are we're going to just talk about them very simply here without going into the details. So this would be coming out of the division. Okay? So if you look at the cell sizes over here, this is large. This is the original adult cell, and these ones are now small because you've taken one thing and you divided it into two. So they're going to be essentially close to half of the content of the original cell's volume or size and whatnot. So this is where we begin the G1 phase. So after a cell is newly divided, they're small baby cells, if you will. And then during this phase, the cells will accumulate uh, nutrients, and they'll perform their functions, but they'll grow. They're going to make proteins, and when they start making proteins, that means they're actually functioning. So whenever they make proteins, they're for a purpose, and those purposes are going to be fulfilled, and so on. Now, these proteins could help it to grow. It can help to do its heart functions or whatever the cell is doing as well. So in G1 phase, the cell is doing everything a cell would normally do. Then it transitions into S phase. S phase, the cell is still doing everything it does, but in addition to that, what it's going to do is going to replicate and copy its DNA. Now, this really only happens in the nucleus, so I guess you can't make any proteins when the DNA is being replicated, but the cell will still kind of behave as a cell, but it won't make any proteins at this place, because you can't make proteins at the same time you're copying it, because in order to make those proteins, you need the DNA. So you can't make any proteins here. Okay? Now, I did stipulate here that it's the chromatid number that increases, not the chromosomes. And we'll talk about that in a little bit. Okay, it's a big thing for genetics when we talk about genetic continuity. Okay. All right, then we transition into G2. Again, this is cell performing its functions. So it's again going to grow. It's going to make its proteins, performing its functions. Okay. Then the cell, depending on whether it's a cell that can replicate in the first place, the cell will enter into mitosis, and the cell will divide into two cells. And then again, when the cell does this division, it can't do anything else, so it doesn't perform any of its functions. Okay. Now, if you looked at the diagram, cells are mostly in interphase because cells are mostly going to be performing their functions. You only want some of them, some of the time, dividing so that you don't lose the function of the organ, the tissue, the organism as a whole. So you don't want to be dividing all the time. And in fact, that's a bad thing. That's cancer. And we'll talk about that later in the course. All right. So going into more detail, I guess, associated with this, is we're going to first talk about the S phase. So this is a simple diagram to represent the S phase. And we start off here with a single chromosome that's made up of one chromatid because there's only one part to it. Okay. Now, when the DNA chromosome gets copied, right, we're going to replicate, we're going to copy it. That means that this chromosome now has two parts to it. Okay. So now this single chromosome, because it's connected, each copy is connected, right? So you can kind of break it here into halves if you want. All right, so this might have been the original one, and this is the copy. Doesn't make any difference. Which one came first? They're functionally the same, and there's no age difference between them. I guess there's no get the sense that this one's older, this one's younger, because they will be exactly copied from one another. So we still consider this a single chromosome, but because it's two parts, we say that there's two chromatids. So after the DNA replication, you start with one chromosome, you end up with one chromosome. But you start with one chromatid, and you end up with two chromatids. Now the website's kind of confusing. It does say that you start off with something like what they call a 2N, and then it writes down that after the division or the replication you have 4N. That is not true. Okay? Now the N is supposed to represent sets of chromosomes, and after this it's still technically the same number because we went from one chromosome to one chromosome. The number of chromosomes has not changed. Now, the 2N comes up in hereditary. That's really where it's used. So we don't really talk a lot about it here. I'm not going to go into the details. You're welcome to look into it, but we're not going to do that here. What I do want to focus on is if we were looking at number of chromatids, is 1, the number of chromatids doubles. So let's say I started off with 11 chromatids. After I perform the S phase, I will have 22 chromatids because the chromatid number doubles, but the chromosome number stays the same. Okay? Now that is somewhat confusing. I know scientists have not chosen the best names for these two things, but we will do our best in deciphering what they mean. Now the centrioles, or sorry, the centromeres, are what hold the two copies together. Okay? And that was the answer for the first question there, centromeres. Okay? Number of chromosomes, as we talked about before, stays the same. So I think in the video it should have been 2N, and after S phase, it should still be 2N, no change. Now if you look at number of chromatids, so if you started off with, let's say, 6, you end up with 12. It's also important to note that each chromatid produced at the end of this copying, or in this situation, they are genetically identical. They're exactly the same. They have the same genes, they have the same information, they have the same structure. They're identical. Now in the video or in the animation, they also showed you some certain checkpoints, and we're not going to go into huge details with these. We'll talk about them a little bit when we get to more things like cancer and stuff like that. For the most part, checkpoints are points along the progression through the cell cycle that the cell undergoes to make sure that the cell is working correctly, okay, before it proceeds to the next step. So essentially what they do is that if the cell is not meeting the requirements of those checkpoints, the cell cycle stops. It could be permanently stopped. It could just be temporarily. Okay, you could have to do more research into that, but they are simply checks to see whether or not the cell should proceed forward in the process, and stopping it from proceeding if it's going to be bad. Okay? And we'll talk briefly about that as we move forward through these different spots. Okay? So the G1 here is pretty straightforward. Is the cell big enough, right? Because you can't take something small and we'll hopefully divide it again into two smaller cells. And so you don't want to keep getting smaller and smaller and smaller. That wouldn't be good. So the cell has to at least be big enough, and then it has to be like a suitable environment, which means that the cell will be able to survive after. If it's happening in your body, generally that'll be a thing here. And it's really just a question of has it taken enough nutrients and performed enough function to get big enough. This is not presented in your animation, but there is some degree of checking to see whether or not the DNA is damaged. I don't provide the process for you or the mechanism for that, but there is some check to see whether or not the DNA is actually damaged during that process of replicating and so on, or if it replicated correctly. Okay? And if it doesn't do that, whatever the checking mechanism is, it will stop the cell from proceeding to the G2 phase. This one here is, since G2 is the step right before mitosis, it's important to make sure the DNA has replicated. So if the DNA has not replicated and is not all correct, then it will not proceed forward. And then also, is it large enough so that when it divides, there's enough cytoplasm and contents for two new cells? And then again, is it a new suitable environment? Okay. Then after we go through these checkpoints, we're going to end up in the actual mitosis itself. Now I have put in here some additional information. I'm going to have about a 30-second delay there because I had to get a drink of water. Well, this dryness is making my throat dry and then cough and whatnot. Okay, so I've done the drawings the same as you, probably the same things that you have. So we've entered out of interphase. We're now going to be dividing, and we've entered into what is called mitosis. And this is a general diagram to show the very beginnings of it, and I've listed some things over on the side here. You can look at these and write them down if they don't match, or you can just see if you did something similar to that, then you're good to go. I've used different colors here just to show that we start off with 2 chromosomes. So there's 2 chromosomes, right? There's chromosome 1 and then 2. And if you had to list how many chromatids are here, there's actually 4 chromatids. Because chromosomes at this point, after they've been replicated from the S phase, will have their copy. So the chromosome number doubles. Okay. Now, pro-metaphase isn't technically a step. There could be pro-metaphase, there could be a meta-anaphase and an anatelophase and so on. So remember, these are like snapshot diagrams, but if you watch the video, it's a process, it's dynamic, it's changing. So this is really just kind of the connection between going from prophase to metaphase, and this is kind of what's happening during that intermediate, I guess, as time is progressing, as one begins to go into the other phase. Okay, so this isn't really a step, it's a transition from one of those stages to the other. Okay. And basically it's just finalizing some of the prophase things, and then the chromosomes are moving towards the middle of the cell, which we call the equatorial plate, or the equator of the cell, whatever you want. And the chromosomes move, they don't just float around, they're moved by the spindle fibers. So the purpose of the centrioles making those spindle fibers is to connect them to the chromosomes, and then from there they can push them, pull them, and then so on. So the chromosomes are being moved by the spindle fibers. Now spindle fibers attach to the centromere, or the kinetochore, I guess, of the chromosomes, and that connection between the spindle fibers and the kinetochore is what helps move the chromosomes. It's not a random movement. So what you'll find in each dividing cell is all of the chromosomes will line up here in a line at this middle spot. Okay? And that is metaphase. So metaphase is when it begins, I guess, when they're lined up in the middle. So I still have my plate here and they're lined up along it, and now you're on those opposite ends and so on. So they line up. I guess there is no transition into anaphase, but as soon as or shortly after they line up, they're going to be pulled apart. And again, it's the kinetochore connecting to the spindle fibers. Now the spindle fibers are being pulled in to the centromeres, and they're pulling the chromosomes along with them. But as they do that, right, your chromosome, which was connected, is pulled apart, and that's why these diagrams are now split. So they did have two structural parts. Each of those structural parts has been pulled apart and is moving towards an opposite end. So these ones are going in this way, and these ones are going in that way. The reason they're drawn like this is because if you're pulling from this point, these would be dragging behind as it moves this way. As it goes this way, these ones drag behind, and so on. Okay? So the chromosomes are split into their two chromatids, or separate chromatids now. So the sister chromatids are moving into opposite ends of the cell. The last technical phase of mitosis is when they arrive at their opposite ends of the cell, and then you get the reverse of prophase happening. So the nucleus reappears, you get the chromosomes starting to disappear, and so on. Okay, so there's this telophase is kind of where they arrive. You'll also notice that there's also this cleavage furrow, or this pinching in of the cell membrane that typically forms in animal cells, and this is the beginning of cytokinesis. I think you can start a little bit earlier than that, and so forth. It doesn't really matter. We'll depict it here. It does happen. So during mitosis, cytokinesis is also occurring. Okay, and we'll talk about the difference between those two things a little bit later. Okay. And then at the very end of everything, going into the G1 phase now, I suppose. So entering into G1 of interphase, you have your two new cells. Everything is back as it should. There's no more chromosomes. There's a nucleus and two regular cells, although they're smaller. And it's important to note that each cell here is genetically identical to each other and to the original parent cell. And the significance of that is, we'll talk about that in just a moment. Okay, so these are genetically identical, they're exactly the same. So cytokinesis is the physical separation. We're not going to go to the mechanisms of how this occurs, but it's the physical separation of one cell into two new cells, which are called daughter cells. So these two cells up here are called daughter cells. They don't say son cells, I think, just because it sounds a little weird, so they went with daughter cells. Okay? So physically into two. Now, technically, mitosis is the division of the nucleus. Okay? Now, you can't have a cell physically separating without each new cell getting a nucleus and the genetics that go along with that, the DNA. So even if you could physically separate a cell, if it was just cytokinesis, you'd have one useless cell because it wouldn't have a nucleus in it. So these two things go together, cytokinesis and mitosis. You don't have one without the other. I guess you could have mitosis without cytokinesis if there's a cell that is multinucleated, but you would never have cytokinesis without mitosis. Or maybe you could technically. Red blood cells don't have a nucleus. That's interesting. But normally, what you have is both at the same time. It's not one or the other. It's always one with the other, and so on. Now, we usually default to calling the whole process mitosis, although collectively, I guess it is cell division that's made up of two processes, mitosis and cytokinesis. But we just say mitosis generally, and that usually implies cytokinesis and a cell is dividing. Now, the significance or the genetic purpose of having this process, or knowing that it's genetic, the reasoning for talking about genetically identical, is that the purpose of mitosis is to repair your body. So if you get a wound or an injury, your body makes new cells to fix it. And those new cells, which are repairing your body, are made by mitosis. You are still growing in your lives, and this is true until you're a little bit older, when mitosis will not allow you to grow anymore. But when you grow, you don't just absorb and have more stuff stuffed into you, um, but you're making more cells. So I don't know how many cells a baby has. Let's say they have 1 trillion, and then by the end of your growing, you probably have like 10 trillion cells. All right. So in order to make those new cells to grow, it's mitosis. Okay, and that goes in also with developing. It also goes in with replacing any damaged cells, and so on. Now, the purpose of why it's genetic material is that if you're doing a reparation, so let's say I hurt my heart, that's not an anatomically correct heart. If I have damage to my heart, I want cells that are being replaced inside the heart to still be heart cells. So the purpose of making genetic copies is that if I have a heart cell that needs to be replaced or a heart that needs to be repaired, I'm going to take a parent heart cell to make copies, daughter cells, that are also heart cells, so that when they're done, they will perform exactly the same function. And that's the purpose of mitosis. If your skin gets damaged, you want new skin cells, do the same thing. You want to get taller, you need more bone cells or things that make bones, you need those same identical cells in order to do that. You want to develop correctly, so that means you're going to expand on the structure of a heart or on the skin or something like that. Then you need more skin or heart cells in the right associated with those organs and tissues to be there. So this is all about making cells that perform exactly like the parent cell, except you have more of them to help grow, replace, develop, and so on. And the only way you can get those exact things functioning the same way is you make each new cell being a genetic copy of the previous cells.